Our first speaker is Dr. Joya Chandra, and she earned her undergraduate degree in microbiology, yay, with minors in both English and Spanish from Louisiana State University. She earned her Ph.D. in cancer biology and immunology from the University of Texas Graduate School of Biomedical Sciences and MD Anderson Cancer Center. And she undertook postdoctoral work at the Karolinska Institute in Stockholm, Sweden, and also in the Mayo Clinic in Rochester, Minnesota. And you'll see all of our speakers have actually been abroad pretty extensively. Dr. Chandra is currently a professor in the Department of Pediatrics at the University of Texas MD Anderson Cancer Center in Houston. She has a dual appointment in the Department of Epigenetics and Molecular Carcinogenesis. Dr. Chandra has published more than 60 peer-reviewed articles, 5 book chapters, and leads numerous NIH-funded projects. Her work focuses on translational pediatric oncology directed towards leukemia and brain tumor therapeutics. Her lab focuses on modifiable behaviors and treatment efficacy, leading clinical research related to diet and nutrition for pediatric cancer patients and survivors. Dr. Chandra is also dedicated to training the next generation of scientists, and she mentors trainees of all different levels. She's been recognized for excellence in education, being awarded the McGovern Outstanding Teacher Award and the UT System Regents Outstanding Teaching Award. We're so happy to have you come and share your work. Welcome to the Hilltop, Dr. Chandra.
Thank you so much, Dr. Bainham, for that very kind introduction. I'm really honored to be here. Thank you to Dr. Hodges and Dr. Bainham and the Lucian Committee for this opportunity. It's really interesting to hear about educators like Brother Lucian that did so many things. It's inspiring, right, to hear about such well-rounded individuals that provided excellent educational opportunities. It's an inspiration to us all, so thank you. So as Dr. Bainham mentioned, I'm at the Children's Cancer Hospital, which is basically a little part of MD Anderson, which is the #1 ranked cancer hospital in the country. Within pediatrics, we have a pediatrics research department, but there's also basic science department. So I have a joint appointment in the Epigenetics and Molecular Carcinogenesis department, as Dr. Bainham mentioned. And I wanted to show you in the Texas Medical Center, the cluster of pink buildings is kind of the core of where MD Anderson is. However, this is what the campus is going to look like, I think, in 2027. I'm on the south campus, which is about 1.5 miles away from here, and there's going to be a beautiful walkway that's in a helix shape with new buildings, so a lot of opportunity emerging in Houston for potential researchers, hopefully like some of you. So I just wanted to highlight the fact that pediatric cancer truly is a global challenge, consistent with the theme of this symposium. And so cancer is the #1 cause of death by disease for children in America. And I'm kind of, I feel fortunate to be giving this talk on September 29th, because September is actually Childhood Cancer Awareness Month. And my role in all of this is to help find cures for the pediatric cancers that are, again, the #1 cause of death by disease for children in America. So across the world, however, this is an even larger problem, and this is an infographic from one of the major funders for pediatric cancer research that's actually led by parents of a child that passed away from a pediatric cancer. There's many different foundations that kind of follow this model, but Alex's Lemonade Stand is one of the most vigorous and kind of broad in their outreach. And so you can see from this infographic that every day, there's over 800 children around the world affected by pediatric cancer. So definitely a global issue that's affecting a good number of children. And so within the US, we have a good idea of how many children are actually affected by pediatric cancer because we have registries, pediatric cancer cases are reported. However, around the world, the true burden of cancer in children really isn't known, especially in lower income countries. And this is from the American Cancer Society, an infographic basically showing what percentage of children they actually capture cancer incidence in. So this is telling you that in the US, over 95% of children and adolescents, we know whether they've gotten cancer. But in other countries, especially in Asia, or other continents rather, especially in Asia, we only know about cancer incidence in 6% or less of children and adolescents. So just the capturing of the information of cancer incidence is not quite there yet. And then once a child is diagnosed, the treatment outcomes can be very different, and the access to treatment is very different. So this infographic is basically showing you that in high-income countries, 96% of patients have access to cancer medicines. In lower-income countries, it's below 30%. So there's disparities in kind of understanding what cancers are occurring, but also disparities in whether they are treated adequately. And so this is a major problem in terms of the number of life years lost of these individuals that are getting cancer, right? Because patients that are adults typically have 15 years of life lost if they pass away, but children are losing many more years of life, up to 71 years of life on average. And these children have the potential to go on to do amazing things. In my own lab, I've had 4 pediatric cancer survivors come through, do research, and then go on to MD or MD Ph.D. programs. And actually, the head of pediatrics at MD Anderson is himself a cancer survivor, so that's really inspiring. He got a bone cancer when he was 14, and he actually does research on that specific type of bone cancer now. So much like adult cancers, there's lots of different types of pediatric cancers. Does anyone know what the most common types of adult cancers are? Any guesses? Colon is one. Lung, breast, and prostate are amongst the top. You don't see those in these lists, right, for pediatric cancer in terms of frequency. So the most common types of pediatric cancers are leukemias and brain tumors. Whoops, and that is actually, they're kind of very close in terms of incidence, but in the US, recently brain tumors barely overtook the incidence of leukemias, whereas this is an infographic from the UK where you can see the incidence of leukemias and brain tumors is roughly equal, but incidence of leukemias is a little bit higher. So there's definite distinctions between the types of cancers that children get versus the types of cancers that adults get, with brain tumors and leukemias being the most common in pediatrics. And then the outcomes from these cancers are very different as well. So this is a graph showing the rate of survival in pediatric patients with acute lymphocytic leukemia, which is the most common type of pediatric leukemia. You can see that and this is divided out by years over time since the 1960s until 2009 of how survival looked for children that got pediatric ALL, or acute lymphocytic leukemia. So if you were a pediatric ALL patient that was diagnosed, kind of when I was born, you had a very poor chance of survival, around 50% or less than 50%. Now that survival rate is upwards of 90%. So there have been major improvements in terms of the care and treatment of acute lymphocytic leukemia. However, this has not evenly been distributed, even in the US, across racial and ethnic groups. And so this graph is showing the 5-year survival of different types of pediatric cancers, but divided by the colored bars by racial and ethnic groups. So what's striking here is that for lymphoid leukemias, which is the same graph that's being shown here, individuals that are American Indian or Alaskan Natives, that's this green bar, have much lower survival rates than whites or even Hispanics or African Americans. So there's definite racial disparities and ethnic disparities currently in terms of outcome for specific types of cancer. And so these are basically oops, I think I'm frozen, unfortunately. These are opportunities for us to understand why these differences are occurring and to actually act upon them. Some of it does have to do with access to care, but a lot of it is actually biological, and that's an intriguing area of study. So our research in our lab actually focuses on the most common types of pediatric cancers, leukemias and brain tumors. And again, just coming back to the need for focusing on these types of tumors is because of the 5-year survival rates. So for patients that have brain tumors, glioblastoma, or a type of brain tumor called DIPG, which I'll tell you more about, we definitely need new targets and better drugs. However, in patients that already have pretty good outcomes, the leukemia patients that I showed you for whom outcomes have really improved dramatically, we need strategies to improve treatments for subsets of patients that don't do as well, and to kind of focus on survivorship. So I like to say that my lab works on blood, on brain, oops, sorry, I'm hitting the wrong button here. Okay. On blood, brain, and balance. Blood, brain, and balance. Energy balance being one of the ways in which we look at modifiable behaviors for as a means of improving treatment outcomes and improving quality of life into survivorship. So first I'll talk to you about brain tumors. So the types of brain tumors that children get are very different from the types of brain tumors that adults get. And this has really been identified by sequencing efforts. So investigators around the world have sequenced a large number of pediatric brain tumors, a large number of adult brain tumors. They've all been high-grade aggressive brain tumors. And there was a really seminal finding that there are mutations in specific histone proteins that occur in pediatric high-grade gliomas. And those gliomas are called diffuse intrinsic pontine gliomas. They're also called midline gliomas, and they're especially deadly. This is an infographic about DIPG, diffuse intrinsic pontine glioma. The pontine refers to the place in the brain where the tumor occurs. It occurs in the brainstem, or the pons. It typically strikes children between the ages of 4 and 11, and the survival rate is less than 1% for these patients. And again, those sequencing efforts defined unique mutations in proteins that are basically the scaffolding for DNA, histone proteins, which are uniquely present in DIPG patients. And so these histone mutations, again, occur in DIPG patients with tumors that are generally in the midline of the brain and in the pons of the brain. So just a little background on histone proteins. Has anyone heard of histones before? Yeah, good. All right. So you guys know that it is a family of proteins around which DNA gets wrapped, and the histone tails are modified by post-translational modifications, things like phosphorylation, acetylation, methylation, different marks. And so this is showing you histone H3.1, where you can see that specific residues have these little marks. So the P stands for phosphorylation, the M stands for methylation, A stands for acetylation. Different marks are occurring on these amino acids. And in particular, histone H3.1 is of interest because the histone mutations that are associated with this disease occur in histone H3.1 or histone H3.3. So, and you can also appreciate from this infographic of how there's more modifications on histone H3 than any of the other histone family members. So we were particularly interested in the residues on histone H3 where there's several marks, acetylation or methylation, and that led us to study a chromatin modifier called LSD1, or lysine-specific demethylase 1. So this is an enzyme that hits one of those marks on histone H3. It was actually the first lysine demethylase that was discovered. This is the ribbon structure of it, and it works as an enzyme to subtract methyl marks on histone H3 K4, so lysine 4 or K9. And it also kind of acts as a scaffold for other proteins to pile onto to affect gene expression. And so we were especially interested in the fact that this particular chromatin modifier has many inhibitors that can inhibit its activity, many of which are blood-brain barrier permeable. So for a brain tumor therapeutic, you need to have a small molecule that can cross the blood-brain barrier. And there was a drug that had been around for decades and had been used successfully in patients actually with depression and bipolar disorder called tranylcypromine. Whoops. And this drug actually also inhibits LSD1, the histone demethylase that we're interested in. And you can see that there's similar structures in LSD1 protein structure with this protein called monoamine oxidase B, which is the primary target of tranylcypromine. So here you have the tranylcypromine chemical backbone, and you can see that this chemical backbone is recapitulated in many different agents, and some of these agents are actually already in clinical trials. So we have studied LSD1 for many years, and we started out studying it by looking at the gene itself, knocking it down, and looking at all of the other genes that were affected. And so when we did this in brain tumor cell lines, we saw that with knockdown of LSD1, immune genes were actually altered. And so this is a list of the immune genes. This was kind of a surprise to us. Because we hadn't really expected that immune genes would necessarily be a target for LSD1 modulation. We expected things like cell death, and you see that in the regulation of apoptosis gene sets. So these immune genes were of interest to us. We looked at those immune genes in a cohort, a publicly available cohort of 247 pediatric high-grade gliomas, and saw that these immune genes were also tracking with LSD1 expression. So this relationship between our histone modifier of interest and the immune genes was retained in a patient population of pediatric high-grade gliomas. So then we actually looked at whether those inhibitors that I listed were also modulating those immune genes. So these are violin plots kind of showing as a group whether the immune genes were altered in different cell lines that were actually patient-derived. So these are cell lines from pediatric DIPG patients collected upon autopsy and generated and generously disseminated by other investigators. And so in these cell lines, we saw that there was a change in the immune genes, so they were also upregulated with these different LSD1 inhibitors. However, this increase in the immune genes did not occur in normal human astrocytes. So that's an important point about selectivity of this approach of inhibiting LSD1, that the immune genes are changing in our cell line of interest, the DIPG tumor cell lines, but not in normal human astrocytes. So since immune genes were changing, this made us think about applying LSD1 inhibitors within the context of immunotherapy. And you'll hear more about this, I expect, from Dr. Triplett later. But there's kind of two ways to think about immunotherapy. One is to actually deliver a modified cell. You might have heard of CAR T cells or engineered T cells. That's a very hot area of immunotherapy. But you can also think about how you can prepare a tumor cell for being recognized by an immune cell. And that's kind of what we were going for with the LSD1 inhibitor. And so we basically did an experiment in a mouse model for DIPG. This was a very complicated experiment because first we have to take a mouse that is immunodeficient. We introduce the brain tumor cells from the pediatric patients that are labeled with a luciferase label so that we can visualize the tumor. That's the lit up part that you're seeing on the mouse heads. And so we have to implant the tumors. We then have to treat with immune cells. In this case, we're using NK cells because that's where our data was pointing us in terms of looking at cytotoxicity enhanced by the LSD1 inhibitors. And then you have to treat with the drug. So this is many weeks of experiments where you first introduce the tumor, treat with your immune cells, and then treat with the LSD1 inhibitors. And what we saw was that there was a decrease when you combined the LSD1 inhibitor, which is referred to as GSK here, together with NK cells that was greater than any of the other individual treatments. So if you treated with the LSD1 inhibitor alone or with the NK cells, the immune cells alone, there wasn't as much of a decrease in tumor burden as you saw with the combination, and that's depicted here in the actual mice, and the numbers are basically reflecting the tumor luminescence, the tumor burden in the mice. So this was actually the work of a graduate student who was a first-generation college graduate, Kevin Bailey, and he published this paper in Neuro-Oncology, and it was highlighted by the editors to kind of be novel in terms of being the first to show that an immune gene signature associated with knockdown of this chromatin modifier is seen in pediatric high-grade glioma patients, and that immune cell killing, specifically NK cell killing of DIPG cells, is enhanced with these LSD1 inhibitors. And this work is being carried on by a current graduate student in my lab, Leah Stitzline, and a master's student in my lab, Faith Hernandez, who's also a first-generation college graduate. So now we'll shift gears completely. We'll talk about blood cancers because that's the other most frequently occurring pediatric cancer type. And so just a quick background on the different types of blood cancers that are seen in children and adults. Blood cancers can arise from different cell types that are either of the myeloid lineage or the lymphoid lineage. You heard me talk about acute lymphocytic leukemia earlier in terms of the cure rates being dramatically improved over the years. So acute lymphocytic leukemia is an acute leukemia that arises, again, in a lymphoid precursor, but acute myeloid leukemia is also an acute leukemia, but it arises in a different precursor. And so acute myeloid leukemia has much worse outcomes than acute lymphocytic leukemia. And then there's also chronic leukemias. These are not as frequently seen in pediatrics. And this is what is being shown in these pie graphs, where you can see the different types of blood cancers that occur in children versus adults. So what do you notice about the biggest slice of the pie in kids? It's ALL. It's that acute lymphocytic leukemia. It's the same tumor type where I was showing you the cure rates have improved dramatically over the years. In adults, though, ALL is a very, very small slice of the pie. So the types of leukemias that children get are very different from the types of leukemias that adults get. However, if you look at the slice of pie that AML is, it's roughly the same in children and adults. So the incidence of AML is not so different between children and adults. What is different, though, is even within AML, the subcategories in terms of the different mutations that occur in AML. And so these are some of the genes that are frequently mutated in AML, acute myeloid leukemia. There's mutations in unique genes in children. So this gene called GATA2 is frequently mutated in children with AML, but you don't see GATA2 in any of these other lists. However, one gene that you do see in common across all of these lists is FLT3. So FLT3 is frequently mutated in AML patients, regardless of whether they're adults, children, or adolescents and young adults. And this FLT3 mutation generally confers a worse prognosis for all of these groups. So that's the type of kind of subset of leukemia that my lab has focused on. And that's what I'll be kind of referring to for the rest of my leukemia-centered talk. So we became interested in how to actually make leukemia treatments better through the influence of modifiable behaviors. And so some of the modifiable behaviors that we have studied over the years have been nutrition, factors that contribute to body weight, and cigarette smoking. So I think this is really empowering for a patient to say that part of the kind of outcome that your drugs might exert can be potentially in your hands. And cigarette smoking in particular, I think is one of the strongest examples of where that may be the case. However, surprisingly, this hasn't been well studied at the molecular level or with mouse models. And acute myeloid leukemia is actually one of the 13 cancers that's most highly associated with cigarette smoking. So everyone thinks about lung cancer, right, as being caused by cigarette smoking, but very few people think about acute myeloid leukemia or even some of these other cancers, I think, stomach, liver, pancreas, kidney cancer as being associated with smoking. Did you guys know that those cancers were associated with smoking? Some of you did. Dr. Bainham did. All right, so acute myeloid leukemia is associated with smoking. And so we partnered with clinicians at MD Anderson to look at a cohort of patients that were AML patients that had that FLT3 mutation that I told you about that's common across children, adults, and adolescents and young adults. And so in this FLT3 AML population, we looked at whether patients that had never smoked versus had any smoking history, so either past or current, had differences in outcomes. And so this complete remission or CR rate was significantly different. This is the p-value. It's 0.001, so significantly different between never smokers and people with a past smoking history. And this is the survival graph kind of showing you that if you are a AML patient with the FLT3 mutation and you have never smoked, your survival is much better than if you had a past or current history of smoking. And so we wanted to study this further in a mouse model to understand what was actually contributing to this outcome. So we have this robot that can actually deliver cigarette smoke to mice. And so we use a model for AML, which is basically human AML cells from a patient that were labeled with this luciferase label that enables us to see the tumor in mice. We first deliver cigarette smoke to the mice that are the smoking mice with this machine. And this machine actually takes research cigarettes. So you can purchase these research cigarettes that are consistent in terms of their delivery of nicotine. And if you buy like Marlboros, then that might not have the same nicotine content as Virginia Slims. So these research cigarettes are a way to kind of standardize the components of cigarettes and are frequently used by the research community. So the research cigarettes are basically lit in these units, and then the mice sit in these chambers and they inhale the cigarette smoke. And so in this case, we had mice that were engrafted with acute leukemia cells, and they were engrafted in mice that had either not been exposed to smoke or had been exposed to smoke. And then we basically followed the tumor burden using this luciferase measurement that highlights the growth of the tumor cells in the mice. And so when we did that, we used two different AML mouse models. We saw that the rate of tumor burden was much higher in both of these mouse models. So here you can see the mice themselves. The non-smoking mice have much lower levels of tumor than the smoking mice, and this is the graphical representation with each dot representing a mouse. Much higher leukemia burden. How about if you treat these mice? So we treated them with a drug called daunorubicin, which is actually used around the world for AML treatment. And what we saw was that in the mice that received daunorubicin, if they were not smoking, there was a decrease in tumor burden. So the blue line represents daunorubicin treatment. You can see there's a significant difference between untreated and treated mice. But in the smoking mice, the daunorubicin treatment was not effective. So the efficacy of treatment was less effective when mice were exposed to cigarette smoke. Well, how about if we got the mice to stop smoking upon leukemia engraftment? This is a very translatable concept, right? Like if we could get a patient to stop smoking as soon as we knew that they had leukemia, would that affect leukemia burden? And it did. So we basically did the experiment of either exposing mice to smoke continuously through the time that they were engrafted, or stopping them after engraftment and then following tumor burden. And you can see that in the mice that ceased to smoke, as soon as we detected leukemia, there was a decrease in these two different mouse models of the leukemia burden. So smoking cessation could be a way to actually slow the adverse effects of smoking on leukemia cell growth. So this was just published in Blood Advances and was the work of a graduate student who finished up last year in my lab, Mary Figueroa, another first-generation college graduate. And we're trying to understand what the molecular mediators are of this accelerated growth and the cessation, because we're basically showing that these chemical signals are either providing go signals or no-go signals for leukemia cells. And that's probably partly through the microenvironment, because remember, we're smoking the mice before we give them leukemia, and that's causing the leukemia to grow more rapidly. So there's a lot of interesting cell-cell communication amongst the microenvironment and the leukemia cells that's occurring here. And we can look at that more carefully with techniques like this, which look at one cell at a time with multiple different antibodies. It's a technique called mass cytometry, which will allow you to look at many different proteins simultaneously on a single cell basis. So smoking is actually a lot more of a global health challenge than I think many of us think about. This was from a series of articles that appeared in Nature just in June of this year, kind of talking about the health impacts of smoking. And again, since this is a symposium on global health challenges, I wanted to highlight part of this infographic, which talks about preventable deaths, non-communicable diseases, and all of the deaths that result from non-communicable diseases are shown in this maroon bar. And of that maroon bar, 14% of them are smoking related. So it's a major contributor to, again, the theme that the symposium is highlighting. And of course, worldwide, it's an even bigger problem. So next, I'd like to talk about a different modifiable behavior. We talked about smoking as being something that can influence treatment outcome. How about nutrition and body weight? So I won't have time to talk about body weight as much today, but I did want to tell you a little bit about our work on diet. And so again, we use mouse models, and then we also have some early clinical trials and some resources for families that are around this theme of nutrition and how to modify nutrition in cancer patients and survivors. So I think all of you are aware that the Western diet is typically high in fat, it's high in sugar, it's high in salt. This is the typical Western diet. And so there's been some really interesting work showing that high-fat diets, for example, can promote AML growth. And this is one of those papers. There's not very many papers, just a couple. We were actually interested in seeing whether changing to a low-fat or a low sucrose, so actually correcting those kind of poor aspects of a Western diet, would influence leukemia outcome. And so in particular, we wanted to understand on a broader basis what diet regimens can help AML therapy work better. And again, we're focusing on this FLT3 mutant AML. So if we kind of veer away and instead of looking at high fat and high sucrose diets, do we affect AML progression? And so the models that we use are basically to do mouse clinical trials. We put mice on different chows. So you can purchase mouse chows that have different amounts of proteins, carbs, fat, and sucrose. And you can basically compare whether AML progression is the same on these different chows, and then whether treatment, such as daunorubicin, the treatment that I mentioned earlier in the context of the cigarette smoking studies, is less or more efficacious or similarly efficacious in the context of these different diet changes. And then we can also look at when we're delivering the food, and we have these cool cages in our mouse facility that are metabolic cages, which can measure the weight of the mouse, how much food they're eating. They can measure their urine, they can measure their feces. You can collect those items. You can measure their physical activity. So it's a monitoring of the mice on a single mouse basis. And you can understand why this needs to be on a single mouse basis, right? Because we want to see in each mouse what the change is. But what we didn't anticipate was the effect of social isolation on these mice. Normally when we do these experiments, so all of the smoking experiments, for example, the mice are housed together after the smoking, so they're together. But in these experiments, they're housed in an isolated manner in these metabolic cages for many weeks, because we want to be able to measure all of these outcomes. And so I think that's an interesting kind of aspect of these experiments that has to be taken into consideration. we do notice that the mice show more signs of anxiety. They're colder. They're kind of seemingly a little more depressed. But we haven't measured these things qualitatively, but it's definitely something that I think we have to take into account. So we basically have taken kind of two approaches to looking at the impact of diet in leukemia patients. We've done these types of mouse experiments that I've described using low sucrose chow compared to high sucrose chow, or low fat chow compared to high fat chow, and then used that chow in AML-bearing mice, treated the mice with chemotherapy, and then monitored leukemia progression. And then we've taken the same macronutrient composition type of diets and seen whether children that are on leukemia treatment will actually eat these foods. And so this work was written up by a research assistant in my lab who was a recent undergraduate graduate from the University of Chicago who spent 2 years in my lab before applying to MD PhD programs, Megan Rodriguez, and she's now an MD PhD student at Columbia. So these are some of the mouse clinical trial results that we've gotten. And so this is comparing the survival of mice that have been inoculated with AML cells and then treated with doxorubicin, one of the drugs that is very similar to daunorubicin that works pretty effectively in AML models. And what we're looking at is survival in both of these graphs at the bottom of the slide. And so you can see that the mice that were living the longest in both of these graphs are the mice that were either on the low-fat diet and treated with doxorubicin, or the low sucrose diet, which is this pea green bar, and treated with doxorubicin. So we would never propose that an AML patient would simply get treated with diet, right? We always want to see whether there's an augmentation of a chemotherapy with the diet, and that's exactly what we're seeing. So again, the longest lived mice in both of these experiments are the ones that got the diet modification along with doxorubicin treatment, which is a chemotherapy. And so we took these macronutrient compositions and tried to match them with actual foods. And so these are the tables of the total fat and sucrose content of foods that were prepared for pediatric leukemia patients while they were undergoing treatment. And so we had a whole menu, a breakfast, lunch, and dinner and snack menu that were either low sucrose or low fat, again, trying to match them to the mouse diets that we used. And we asked the kids whether they liked or disliked or felt neutral about the foods that we were preparing for them. And so the majority of them liked the foods. These were children within the ages of 7 to 17, and so kind of a broad range. And so all of the recipes received either a like or neutral rating by 67% of the participants. And some of the things that we got suggestions back on were whether we would consider changing the fruits and vegetables that were in some of the recipes and removing spices. Some participants were more sensitive to the use of spices. So this is kind of something that is preliminary data for designing a menu that is more closely matched and acceptable to pediatric leukemia patients in the future. So in general, we're kind of trying to understand how to define these diet parameters. What are the actual macronutrients that we need to modulate in order to affect chemotherapy outcomes? And there's been a lot of interesting work looking at very specific amino acids and how specific amino acids, for example, methionine, can affect leukemia and other cancer chemotherapies' efficacy. But we really want to focus on whole food deliverable diets because those methionine-specific diets require you to consume generally actually methionine-free food, but it's not really food. It's more like a shake that you have to eat consistently for a period of time, and that's kind of challenging, especially when you're undergoing cancer treatment, to say you can't enjoy whole foods. So we're trying to kind of integrate whole foods into this approach. We also have to figure out a way to deliver these diet interventions, and we've done some work in this space with a research dietitian that has provided counseling that suggests specific foods, suggests specific recipes. And then we've developed an electronic cookbook that I'll tell you more about in a moment that is a kind of source for some of these recipes. And then the really challenging part is actually maintaining and practicing these healthy diets through cooking and actually changing family behaviors, because especially for pediatric patients, there's the ecosystem, the family ecosystem of cooking and food preparation. We've looked at that a bit as well through the use of cameras, which have been used in food preparers of families. So I've had a former graduate student that went into the kitchens of families that had a pediatric cancer survivor and actually filmed how food was being prepared and whether they were using healthy cooking practices. And it was always very interesting to see what was reported versus what the camera captured. And she actually did the experiment on me first, and I failed. Like, I reported out that my cooking practices were much healthier than what was actually captured. So in general, we're kind of interested in diet as well as other modifiable behaviors. I mentioned the smoking part. We're also collaborating with folks in our department that study the impact of exercise and physical activity. And we're interested in circadian biology too, because all of these are modifiable behaviors that have potential to influence the course of cancer outcomes as well as cancer treatment outcomes. So for the patients that do survive, and I showed you that for ALL patients, that fraction is quite high, upwards of 90% now, thanks to all of the new treatments that are being delivered and developed. However, survival is not always as rosy as we would think. And again, this is another graphic from Alex's lemonade stand. And in particular, I wanted to highlight the fact that 2/3 of childhood cancer patients will have long-lasting chronic conditions from their treatment. And so this kind of future of facing a life-threatening illness is significant. And so we also have to think about the ways in which the treatments we're delivering can affect bystander cells and have long-lasting impacts. And actually, the drug that I used consistently in this talk, doxorubicin and daunorubicin, they're part of a class of chemotherapies called anthracyclines, which are known to damage cardiac cells. And so the consequence of this cardiac damage is that many of these patients have early mortality, but it doesn't happen until 10 to 15 years or more after they've already won their cancer battle. And so that's what these graphs are showing you, that the survival of patients that are childhood cancer survivors, so the childhood cancer survivor population has a much lower rate of survival if you look at time since diagnosis in years compared to age-matched controls. And you can see that as more years pass, the bigger this disparity becomes. This is uniform in males and females. And so this is a major issue. Why are these children going on to kind of overcome their cancer, but then as adults, dying earlier? And so this is a field of extensive study, and some of the reasons for these early deaths in cancer survivors are because of recurrence of the original tumor. That's the most frequent cause for deaths. Second, malignant neoplasms. So sometimes they get second cancers. This could be, unfortunately, due to the treatment that they got in some instances. So some of the agents that are used, radiotherapy, some chemotherapies, they cause DNA damage, which are kind of precursors for causing second cancers. But there's also a host of other reasons for early deaths that include cardiac, pulmonary, even ocular late effects. And so that's what these very busy graphs are showing you, but I just want you to focus on the colors. And so these are graphs showing the cumulative burden of individuals across different ages for different types of conditions, health conditions that are faced by childhood cancer survivors. And so the maroon bars are cardiovascular problems that emerge in survivors. And so as you can see in these bars, the maroon bars are the biggest of all of the bars. So cardiovascular issues are faced by the greatest percentage of childhood cancer survivors amongst the chronic health conditions. And then second is this kind of green bar is most highly represented across the groups, and that is referring to endocrine issues. I think you'll hear more about endocrine issues and diabetes in our next talk. And so these are some of the kind of late effects that we have to balance our chemotherapeutic regimens with, and also kind of think about how we can deliver the treatments for pediatric cancers with those late effects in mind. So this is something that we want to and need to integrate into our models of cancer treatment. So we have to think about the mouse models that should be used and whether the diet regimens are relevant to human diets. So in terms of the mouse models to be used, we know, again, that these late effects are occurring in patients that are treated while they're young but develop when they're older. So when we're designing our mouse models, we have to deliberately choose younger mice that we can observe these late effects in. And so that's kind of a thing that you have to consider when you're designing the experiments, and it's not frequently integrated into survivorship mouse studies anyway. We also have to think about the diet regimen relevant to human diet. So I showed you in the context of the mouse chow versus the human recipes that we're preparing, kind of matching macronutrients. But unfortunately, there's all kinds of other fillers in mouse chow that humans would never eat. And so that's a limitation of really saying that this is perfectly matched to a whole food diet. But I think by trying to match as well as we can the macronutrients, we can at least get some guidance, and then it's up to us as basic scientists that are kind of trying to look at biomarkers for diet change to try to match those across mouse and human models. We also have to think about different patient subsets and age ranges to be studied, and how the diets can be delivered in a manner that accommodates symptoms and preferences. This is a pretty big issue, so this is something for all of you to think about. You know, what are the types of symptoms that you've heard about that cancer patients may experience? Nausea, vomiting, lots of GI issues, right? And so we have to think about foods that can be delivered that actually accommodate some of those symptoms. And then you saw from the early slides that I presented that there's lots of different subsets of patients, and the tumor types occur in different age ranges. So the foods that we design for younger children may not be applicable for older children, and then more importantly, the diet regimens have to be examined in the context of some of those very specific cancer types. So the studies I showed you are just in one very small subset of acute myeloid leukemia patients. We don't know whether we'll see the same effect in acute ALL models. However, we're doing the trials primarily in acute lymphocytic leukemia patients because that's the most frequently seen type of cancer in the clinic. So we have to kind of try to better match the patient models with the mouse models in terms of tumor types. And then how can families and their food and cultural and social aspects be accommodated? These are all things that have implications into survivorship because we want these healthy habits to kind of persist into the sphere of survivorship. So when we started our diet studies and we started kind of thinking about preparing foods, the issue of kind of maintenance and having a repository of foods came into our minds. And we decided we were going to try to design a resource for kind of promoting healthy cooking and eating. And so we kind of on a grassroots level started collecting recipes from employees, from the community, and we designed this electronic cookbook, which is freely available. Anyone can access it at mdanderson.org/recipes. It's got the ability to look up recipes based on symptoms that might be experienced. It's got recipes that that can be sorted based on time needed to make the recipes. There's also lots of tip sheets that are available in English, Spanish, Mandarin, and Arabic. And there's unique aspects of this website relative to other websites that are linked to academic centers. So we have over 770 recipes. There's a child and family focus. You can search by symptom needs or by nutritional needs. And we've published a couple of papers on this resource. So in summary, many of the behaviors that are often associated with cancer prevention, we also need to think about in the context of cancer treatment and survivorship. And so this is just some of those behaviors that I think many of you are familiar with. And I think each of these is a field in and of itself in terms of thinking about the things that I mentioned, figuring out the exact way in which these behaviors should be delivered, figuring out what molecular biomarkers could be of actually making that change, delivering those interventions in patients and in survivors. So before I end, I just wanted to plug very quickly how important it is to get hands-on research if you're interested in doing this type of work. And so I wanted to just highlight the fact that, as Dr. Beta mentioned, I was an undergrad at LSU in the microbiology department, but I had no idea of what research really was. I kind of wasn't sure if I wanted to go to med school or what else there was beyond med school. But it was really a life-changing experience to do summer research at MD Anderson after my sophomore year. Whoops. And so that's what I did. I spent a summer in the pediatric department at MD Anderson actually doing clinical research. So I was going through charts of patients that had received radiation therapy and seeing whether their test scores were different from age-matched controls. And what do you think I saw? It was different, right? So that was my first clue into, wow, treatment can actually affect patients in all kinds of ways. And so after I finished my undergraduate degree, I still wasn't really sure of what I wanted to do, and the research experience I had was not lab-based, it was clinical research. And so I spent a year after I got my degree as a research assistant at Baylor College of Medicine, working in a lab doing ovarian cancer research. And I had already been accepted to the Graduate School of Biomedical Sciences. I deferred admission because I really wasn't sure whether I liked doing hands-on research. But I liked it, and I joined the Graduate School of Biomedical Sciences, where Dr. Beta mentioned that I got my Ph.D. So now I actually oversee a summer program at MD Anderson, which offers hands-on research experiences. And I encourage any of you that are interested in applying to apply. I think the applications hopefully will open in December. It's called the Summer Program in Cancer Research. And the website is here. There's actually several summer programs at MD Anderson. This one in particular is focused on doing work at the bench and on epigenetics in particular. So I just wanted to thank the wonderful members of my lab that have contributed to this work. I identified many of them, and then our long list of collaborators and our funding sources. And with that, I'm happy to take questions. Thanks so much for your attention and for the opportunity to be here.